That's beautiful.
Hi, everybody.
You ever heard of that? That's why that being so simple.
I'm Oliver Sutter, and I'm the director of Martin Pastor University.
Do what God tells you
It
to do.
is my pleasure to introduce three of our students, and they will be talking about what they plan to be researching for their honors project in the coming year. We have three juniors with us. We have Brian Nguyen, Liz Paul, and Nguyen. And I will introduce the first one here. This is Brian. He's going to be talking about gene function characterization of four toxic genes in Mycobacterium smegmatis bacteriophage LeBron. Did I get close? Fantastic. So take it away, Brian, and then I'll come back up and I'll introduce Liz.
Hello everyone, my name is Brian Nguyen. I am a biomedical science major in the pre-med pathway, and I'm honored to be presenting my scholarship presentation to you all. My proposed project involves the characterization of four toxic genes in Mycobacterium smegmatis bacteriophage LeBron. Now, when you look at that title, you might see a bunch of sciencey words just thrown together. Perhaps maybe one or two words might jump out of you. If you're familiar with basketball, it might be LeBron. For others, it might be that really long Latin name that no one can pronounce, Mycobacterium smegmatis. But sandwiched in between those two is this word, bacteriophage, which is really important. But before I get to what a bacteriophage is, I'd like to outline what I will be presenting on. So first I'm going to start with an introduction to what a bacteriophage is and how it operates. And that will lead into the discussion of why is it important that we study bacteriophage. Then I'll be talking about my personal interest and things that I've done to prepare myself to undertake this project. Then I'll be talking about the methodology that I'll be utilizing in order to complete my project, my plan for the year of research, and then finally I'll end with a conclusive summary of my overall project. So, what is a bacteriophage? Simply put, a bacteriophage is a virus that infects bacteria. Bacteriophage, or phage for short, are actually the most abundant biological entity in our world, with 10 to the power of 31 particles existing in the total phage genome. If you look over to the side, there's a model of a typical bacteriophage, and if you notice, it consists of this protein head that is connected to this tail, and at the end of the tail, there are these tail fibers. And these tail fibers are really important in the way that it infects bacteria. So the way it infects bacteria is that it attaches itself to the bacteria using these tail fibers. It penetrates the bacterial membrane, injects the phage DNA, which is housed in this protein head, into the bacteria. And once it's inside, the phage DNA will utilize post-reproductive machinery in order to replicate itself, create more viral particles, and ultimately lyse the bacteria to release these out to infect more. So the particular bacteriophage that I'll be researching on is named LeBron. LeBron has 123 protein coding genes in its genome, which is relatively large for a genome of this nature, and its host bacteria is named Mycobacterium smegmatis, the importance of which I will be getting into later. So why is it important that we study bacteriophage? Well, they have many useful applications in our world. They're often used in agriculture, biosanitization, and biopreservation, all for a very similar purpose, and that is to prevent bacterial growth. But as a pre-med student, I'm interested in their medical applications, and an area of increasing interest is something called phage therapy, which is the use of bacteriophage in order to treat bacterial diseases. So this is especially important in an era of increasing antibiotic resistance. So normally if you're diagnosed with a bacterial disease, you can treat it with antibiotics. But due to the overuse of antibiotics in recent years, that has led to a rise of bacteria that have gained a resistance to this. So one possible solution is to use bacteriophage to kill these resistant bacteria. Now I mentioned that there is a particular significance in studying a phage that infects Mycobacterium stigmatis. And that is because it is biologically similar to the bacteria that causes tuberculosis, which is called Mycobacterium tuberculosis, very similar sounding name. Due to this biological similarity, as well as NSMAG's fast replication and relative non-pathogeneity, it serves as a really good model in order to study tuberculosis and other similar bacterial diseases. So why am I partaking in this research project? Well, I have gained an interest in molecular biology thanks to my participation in the C-Phages and C-Genes program. So these are undergraduate research-based courses that give biology students an opportunity to gain hands-on experience in a lab setting. So in C-Phages, we had the opportunity to isolate our own bacteriophage from an environmental sample, and then we also had the chance to assign gene functions to particular genes of phage genome using bioinformatics and comparative analyses with other similar phage. However, there were some genes that we just couldn't assign functions to, and that was just because of a lack of data. So we had to figure out other ways to characterize these gene functions. And that's what we did in C-Genes. So in C-Genes, we started off by creating a library of phage genes that were cloned into this P extra plasmid. Basically, a plasmid is an external piece of DNA that bacteria have the capacity to take in and use. So this entire process involved an amplification of phage genes, and then transforming bacteria containing the plasmid to create a culture that we can use in later tests. So after we created this giant library, we were able to do something called cytotoxicity assays. Basically, we grew bacterial colonies on the agar plates, and then we tested to see the effects of phage gene expression on bacterial growth. If the bacterial growth was inhibited in any way, we can label these genes as toxic. So these are two examples of some cytotoxicity assays that we did. On this side, you can see an example of a non-toxic gene. If you focus on the three columns to the right, you can see that even as we expressed the phage gene, there was very little effect on bacterial growth. Therefore, we can label it as non-toxic. Contrast that with this image, again focusing on the three columns to the right, we can see that as we express the phage gene, it caused a significant decrease in the amount of bacterial colonies, and therefore we can label this as toxic. So this cytotoxicity assay is really important, they're really helpful in characterizing gene function, but it only illuminates one aspect of the gene function. We still don't know how exactly these genes are being toxic to the bacteria. We don't know the mechanism of action. And that opens up a lot of opportunities for further research in gene characterization. Which leads very nicely to what I'll be doing for my project. I'll be selecting particular toxic genes in phage that run and performing a protein-protein interaction assay. Basically, I'll be seeing if the phage gene products interact with any proteins in the bacteria. So the entire process for this is a little bit complicated, but I'll try my best to simplify it. The main star of the show is this plasmid, which is named P2H alpha. And this plasmid contains particular components that will allow us to test for this interaction between the phage and the bacteria. So it will contain this gene, phage gene insert, but that's the gene of interest that we'll be testing. And if you notice, it is connected to this purple part, which encodes for the alpha domain of an RNA polymerase. RNA polymerase is an enzyme that is responsible for the first step of protein synthesis. So if you transcribe this entire region of the plasmid, you create this RNA polymerase complex that contains this alpha domain connected to the phage gene insert. So we can put this plasmid into specially made bacterial cells that have a special reporter gene, and this RNA polymerase can transcribe that reporter gene, but it is only functional if there is an interaction between this phage gene insert and the host bacterial protein. So this interaction will stabilize the RNA polymerase and allow it to transcribe the reporter gene. This reporter gene causes the bacterial cells to appear blue. So if there's bacterial growth and it appears blue on the plates, then we can confirm that there has been an interaction, and then we can work on identifying what exactly that protein is that is interacting. So here is the genome map of H. lebron, and while it is not as big as like the human genome or animal genome, it's still a pretty sizable amount to be studying within a year. So I had narrowed down my selection to these 4 genes. Genes 35, 50, 59, and 66. Now this is a tentative list as we're still testing for toxicity in other genes, and they could potentially be candidates for interaction assay, but for right now these are the 4 genes that I'll be doing my research on. So this is my scheduled plan of research for the next year. Right now we're still doing some cytotoxicity assays in order to test toxicity in the rest of lebron's genome and potentially use them in our protein interaction assay. In the fall, I hope to be cloning these phage genes into the P2H alpha plasmid that I had talked about, and this will involve a very similar procedure that I had discussed earlier using PCR, bacterial transformations, and putting that plasmid into a bacteria to create a culture that we can use later. In the winter, I hope to be doing the interaction assays for all 4 of my genes, and in the spring, I hope to be at the part where we are identifying the host bacterial protein that had interacted, as well as we do any tests that need to be completed. And hopefully by the end of this quarter, I'll have all the data needed to present my full project. So, just as a summary of my overall project, I'll be doing an interaction assay to identify protein interacting partners between the phage gene product and the bacterial host protein. This is particularly helpful in further characterization of phage genes with no currently assigned function, and it helps us understand the biological importance between the interactions between the bacteria and the phage. Ultimately, this research holds applicative value in the field of medicine, as we could potentially use these toxic genes in treating bacterial diseases through phage therapy. We can create therapeutics and phage-derived antimicrobials in order to target specific bacterial proteins to treat these diseases, as well as ensure that only the most effective phage are being utilized in the innovative treatments. Before I end my presentation, I'd like to thank my sponsors for my project, Dr. Diaz and Dr. Nithaya. Dr. Diaz focuses on virology, and Dr. Nithaya focuses on microbiology, so they are very knowledgeable on this topic and have served as great mentors as I partake in this research. I'd also like to shout out 2 members of my research lab. First is Elizabeth Paul. You'll be meeting her soon enough. She's the person who introduced me to the lab and showed me around the lab space, and is a very close friend of mine. The other person is my lab partner, Erica Jane. Another close friend of mine, and I couldn't be happier to do research with anybody else. And with that, I'd like to thank you all for listening to my presentation.
Excellent job, Brian. Thank you. Does anybody have any questions from the students? Dr. Garrigus, I see the wheels turning.
I wanted to see if anybody else wanted to go first. First off, all 3 of you, amazing presentations. I mean, I think
I'm super impressed with the planning that you've already done and the way that you've thought ahead about what you need to do and your goals. I just think you're all set up for success. It's just, now you just have to do the work, right? But really kudos, and I'm so excited that alumni are here to see you, because I think they must be bursting with pride, as your faculty are, to see you. My question has to do more with methodology. So our first presenter, and I'm sorry, Brian? Right, okay. So you're coming at your project in the most traditional way. You're a major in the field and your research is in that field. So my question is more directed at the second 2 students. Our third presenter, I'm sorry, your name?
Amber.
Amber, what is your major?
I'm also a
That's what I thought, okay. So what I'm curious about for Amber and for Liz, right? So talk to me a little bit about how you sort of, in a sense, had the imagination to switch from your major to the discipline that you're doing your research in. And I'm tickled that you've got faculty in the room that might also want to back you up on this. But I'm just curious, how do you plan to sort of scaffold your knowledge and skills in a discipline that you have not been spending the first few years of college studying?
And don't worry, Brian, I have a question for you.
Sure, I'll go first. So, for me, I think a lot of it had started actually when I was a kid. And so at my church, we have this thing that kind of started in the past 20, 30 years, where all of the kids that are in our church, in order to kind of get into higher ranking positions in the church and to get married, it's not really set in stone. But for a lot of those positions and whatnot, they actually have us go through Sunday school for 10 years, and we take an exam at the end of those 10 years. That is not only talking about our religious doctrine and Bible stories and whatnot, but it also talks about church history. So that was always my favorite part. It was my friends, they all hated it, but I was in love with seeing that. And so when I got into college, I did more research in the biology department, and I got really familiar with doing a lot of research for a lot of the research papers that we've had to write. Brian knows they're super involved. And when I got to the class with Dr. Ferreras, I had realized practical theology is very, very cool. And I wanted to always kind of learn more about my heritage in that sense, because I think when you grow up within the church, you're kind of told this is what's happened, and usually it's not from an unbiased perspective, right? So a lot of things that they say have happened have either not happened or not happened in the way that you thought. So I was thinking about that. I talked to Professor Sutter for a long time. We had weekly meetings that were hours long, but we would talk about this, and finally I just asked him if it was strange that I was going to do this, and he was very excited for me to do it. So I asked, and yeah, that's how I got into this.
I think for me, what was most important to me, as soon as I heard about the Honors Scholarship Project, in freshman year, I was thinking to myself, the two big things for me were feasibility and then applicability. My goal going into figuring out a project was, I want something that I want it finished. When it's finished, I want it to be able to have an impact on the world, even if in just a small way. I wanted something that could directly be taken and used for anything from policy recommendations to this is how these people are, maybe these are ways to help them, etc. And that interest, at first I was actually thinking about doing diabetes or something like that in the Inland Empire, because it's also a huge issue. Because as a bio major, I'm still very much concerned with health and stuff like that, so I wanted something in regards to that field. I wasn't too sure. And then it was the UHNR 314 with Dr. Mallory and Dr. Fidelis that kind of cemented it for me, because we talked about the people, and we talked about the communities, and we talked about all these different problems, and I just saw there was also sort of a gap in the literature, especially in regards to our local communities. And so I went to Dr. Gonzalez, and then she connected me with Professor Patel and Dr. Vega, and it kind of just took off from there. That's all. Thank you.
I actually have a question for Brian. Brian, on one of your slides, you had the grids of which genes were toxic and which ones were not. And I noticed on 2 it was very obvious that there was lack of growth, right? But on the third one, it seemed very similar. And so I was wondering, what is your rubric on the grid for how many grid spots become a toxic gene versus how many are qualified or categorized as non-toxic? And thank you, Amber and Liz. Your presentations were also fascinating.
Thank you for that question. That's actually a really good question. So part of the reason why there are 3 colonies is because we have 3 samples. So when we grew our bacterial colonies, we selected 3 isolated colonies to test for toxicity to just ensure that the results that we get are replicable. So because of the presence of those 3, we can compare all those 3. So the third one might not look as toxic as the first 2. But because of the first 2, we can probably conclude that it was toxic. And for toxic genes, what we have to do is we repeat the entire cytotoxicity again to verify that, okay, it is actually toxic. So I didn't show it, but we had another test for this, and it did confirm that it was toxic. Another thing is we use that to test the level of toxicity to see, like, oh, is it just like smaller colonies, or is it actually decreasing the number of colonies? So, yeah, very good question.
Well, I just want to congratulate all 3 of you. You were amazing as freshmen, and you're still amazing. I have a question for Brian, but first let me just say I heard a rumor that at the University of Memphis there's a new bacteria that can resist LeBron. It's called the John Morant bacteria, and in the next week it's going to defeat in 4 out of 6 contests. So we'll see how that goes. Brian, there's almost always a fascinating story behind these bacteriophages and where they're located on campus. You know, I remember at one point a couple of years ago, the theory among the biology students was that the sludge down in the ponds at Riverwalk was the best place to find them because of all the duck nitrates that ended up in the pond. So where was the LeBron? I mean, is there a story behind the LeBron bacteriophage?
So, LeBron is actually not a Lacera bacteriophage. It was discovered in, I believe, in Virginia, 2008, which is much before the C-Phages program started here. Also, I did hear rumors that the sludge is very good. But yeah, so I don't really know too much other than where it's found. It was in Virginia, because C-Phages is a program that extends to a bunch of universities, not just Lacera, but a whole amalgamation of these universities that combine together to work on this research. And I assume the name comes from the fact that the person who discovered it might be a big fan of LeBron. But that is as much as I know about LeBron in terms of his origin.
Thanks, Brian. Are there any other questions? I have one for you, Brian. You said you picked out 4 specific genes. Why did you pick those? Is there a story behind that?
I was expecting someone to ask this question. So each gene has a particular reason why. So if you remember, my genes are 35, 50, 59, and 66. So gene 35, the reason I picked that was because on the genome, it is located right next to a gene that encodes a tyrosine integrase. This is a protein that is involved in the DNA insertion of the phage DNA into bacteria. So genes that are located next to each other generally are involved in the same cellular process. So we expect, even though we don't know the function of this gene, of gene 35, we can expect that it is involved in DNA insertion, and if it does, it must have some interaction with the bacteria in order to penetrate the bacteria. It's also interesting because that gene in particular is only found, it looks like it's only found in certain bacteriophage, which means it must have an auxiliary role of some sort. And so that points to some point of interaction. We're going to skip 50 for now, I'll explain that later. For 59, through the information that we do have on it, it looks like it can be a transcription factor and transcription factors basically help stabilize interactions with DNA binding proteins. So if it is indeed a transcription factor, then we would expect that there would be some interaction with these DNA binding proteins. Gene 66, we picked that because it looks like it is related to a gene in another phage that encodes for something called the N-terminal phage ribolosome organizer. Very fancy name, but all it really does is it is part of a protein complex that is also involved in DNA replication. So DNA protein complex meaning a combination of all these proteins. So if this gene is related to that, we would expect that it would be part of a similar complex and therefore is interacting with the host bacterial protein. Now we get to 50. 50, we have not a lot of information about it. It's not next to any important gene. It's not like, we don't know if it's a part of any cellular process that is particularly interesting. The reason why we picked gene 50 was because my lab partner felt this very strong connection with this gene number in particular, and we were like, all right, let's do it.
Very scientific. Okay.
Fantastic. Are there any other questions?
Every researcher is needing some sort of funds for the laboratory support, and how did you go about obtaining that?
So the research that I'm doing, it's a natural progression of the C-phage C genes programs that I have been part of and that I discussed, and those are sponsored by this group called HHMI. So when we actually when I said that we identified the protein, the bacterial protein that interacts, right, we're actually sending it to them so they can do all the sequencing and send us back the information about that. In terms of the lab materials that we'll be using, I believe it is funded by the Ross Year Biology Department, so everything that we have within the lab is all sponsored by the Biology Department. So it's very expensive equipment.
Donations are accepted.
Does anybody else have any other questions? I maybe have one for Amber. I think it's interesting that you're talking about food deserts and you picked out cities in an actual desert. Do you anticipate or have any idea that maybe there'll be something to sort of learn about what happens in the Mojave Desert, where something where it's very like the logistics of the inputs to live out there, you have to bring things to where you're going to live. That there's maybe anything to learn from that applies to more traditional suburban environments like where we are right now here in La Sierra.
That is a very good question that I've never thought about, so I'm speaking as I go. I think a lot of this will be covered as I do more thorough literature review, because for the most part, in regards to the actual area that we've decided, it's been more informal interviews, me talking with, say, family members or community members that I know there. And as such, I don't want to answer you with a very hard yes. I think that there's a lot of parallels you can draw between the Mojave Desert and the high desert area. The big thing too is that so food deserts, they're like urban ones, and there are rural ones, and the sort of definitions for them, according to the USDA, really depend on the area and the population density and such, because a lot of the sort of parameters for what defines a food desert are determined based on, like, say, opportunities per square miles and such. So it's, I don't know how much I will be looking into, say, like the Mojave Desert or something. I don't know how comparable it could be. I will get back to you by next year, though.
All right, cool. Are there any other questions? I think if there's no other questions, let's give our presenters a hand. I thank you all for coming out today. Really appreciate it.